Hi dear students, welcome back to the staining techniques, and previously we had discussed about the different types of stains which we have, the acidic as well as the basic stains, which mainly have positively charged as well as the negatively charged groups within it, and they mainly have ionic interactions with certain certain parts of the particular microbes as well as the different things which we view under microscopy. And but I would also like to add on to it, we also do have certain covalently binding dyes. These are mainly dyes which form covalent bonds or which have certain solubility characteristics. Okay, now they mainly, for example, we have fulgen stain. Okay, where you can have fulgen staining procedure where the DNA is being stained. Okay, uh, and you can see that the staining component of the Schiff's reagent will covalently attach to the deoxyribose sugars of DNA. That is a case in which a stain is having a specialized binding character with one particular ingredient, or sorry, one particular component of the particular cell. And also sometimes you can have Sudan 3 black stains also. They mainly stain lipids because the lipid is soluble, and but they does not dissolve in the aqueous portion of the particular cell. So sometimes you have certain specialized stains also which would have covalently bind to what? Specialized ingredients which have been, or specialized components which have been present in the particular cell. So now that we have the ionizable as well as the uh, ionizing based dyes as well as the covalently binding dyes, let's move on to the different staining techniques. If we go to consider the types of staining techniques, you have basically two types: a simple staining technique as well as a differential staining technique. As we had mentioned earlier, in a simple staining technique, you use a single type of stain to visualize the microbes, and you can either see the morphological shape as well as the arrangement of the bacteria or the microbe under study using a simple staining technique. But in differential staining techniques, you mainly use two contrasting stains which could be uh, easily distinguishable, and between using two stains, after putting the first stain you will use a decolorizing agent so that the stain taken up by certain organisms will be removed, but some microbes within it will not get decolorized. And then when you put the second stain, certain other microbes which get decolorized will take up the second stain. So this is a differential staining in which you use two stains. So uh, using differential staining technique, you can distinguish different properties of the bacteria. Now, sometimes you can classify the organisms by using the gram staining technique, or sometimes you can identify certain specialized microbes using certain acid fast microbes. We'll be explaining in that what is it in the coming sessions. That is called the acid fast staining. Or sometimes you can visualize certain specialized structures of the microorganism like the spore or the capsule by certain specialized staining techniques. Now sometimes you can view the flagella, that is also using by flagellar staining. You can use visualize the spore of the microbe by using the spore staining technique. You can view the capsule by using the capsule staining technique. So all these differential staining techniques mainly involves two different stains enabling us to distinguish various properties of the different microbes under the study. So let's move on. Now, what is a simple staining technique? As I mentioned, we use a single stain over here, and normally the basic stains, crystal violet, methylene blue, basic fuchsin, these are the ones which we use. And for the process, you prepare your bacterial smear, air dry, heat fix it, and onto the heat fix smear, you will put your particular basic stain and allow it to react for 1 to 2 minutes depending upon what stain you are using. And then after keeping it for a period of time, you wash it under running water, you air dry the glass slide and view it under the microscope either at 10x, 45x or 100x. Of course, you remember in 100x we use only oil immersion. The oil immersion microscopy is being done in the 100x in the 100x mode. Okay, so let me ask you, in the first case, I told you in the simple staining technique, we use a basic stain. I guess you recollect. We are using here crystal violet, methylene blue, basic fuchsin. These are all basic stains. And basic stains are the ones which what bind to the microorganisms because they are the surface of the bacterial surface is being found to be negatively charged. Now my question is, what if we use a acidic stain for simple staining? Okay, what is the speciality of an acidic stain? Acidic stain does contain negatively charged molecules. So what would be the result if you take a microorganism and use an acidic stain for it? Now what this would be the result. That is, you get a chance of negative staining over here. The dyes, of course, which we use here could be Indian ink or nigrosin and all that. And as a result, what happens, since the bacteria is being found to be negatively charged, and since you are using a negative molecule, negative charge containing stain over here, what happens? The bacteria will not take up the stain. Being negative and negative, so it will not attract towards each other. They get repelled with each other, and only the background of the glass slide gets stained. And as a result, the unstained cells will appear as bright objects against a dark background. So this is a case of negative staining. So within the simple staining technique itself, when you use a single dye, depending upon what dye you use, you will either get a negative staining or a common staining technique in which the microbe, as we mentioned here, see, it is a microbe which is getting colored over here, but the background is being found to be clear in nature. So now we're going to talk about the differential staining technique. The most frequently used differential staining technique is a gram staining technique. It was being named after Danish bacteriologist Hans Christian Gram in 1880. And it mainly helps us to distinguish between gram-positive and the gram-negative organisms uh, among the bacteria. Okay, you might have a doubt what is a gram-positive bacteria and what is a gram-negative bacteria. During the gram staining procedure, you mainly use two different stains. One is crystal violet and one is safranin. All those bacteria which appear to be purple in color or which takes up crystal violet is considered to be as gram positive, and all those microorganisms which take up the second stain safranin, which is pink in color, is considered to be as gram negative in nature. So if you go to consider the whole pool of microorganisms, some of them take up with safranin, they are gram negative, and some of them takes crystal violet. Hans Christian Gram, he noticed that this character, that is, when he was using two different stains, some of them took like this and some of them took the pink color. So he used this particular technique of gram staining to classify the microorganisms. Okay, I'm talking about bacteria specifically. The bacteria can be classified into gram positive and gram negative based on the gram staining procedure. So let's see what is the procedure. If you go to consider the steps, the first you take as a common procedure, you prepare a smear of your bacterial cell, you heat fix it, and after that you first stain it with crystal violet. That is the first stain which you put, and put the glass slide for 1 minute. And after 1 minute, you just water rinse it so that the stain goes. And as a result, what will happen? All the bacteria will take up crystal violet, because when you, as I told you, all the bacteria is considered to have a negative charge. So they will take up because crystal violet is a basic dye. What will happen is a basic stain. Here, all the bacteria on a glass slide, if it has two different types of bacteria, will take up the crystal violet and they will appear to be purple in color. And after that, you add another thing called the iodine. Mordant. Is something which will enable this the crystal violet to remain more attached onto the particular what? Onto the bacterial cell. And you put the grams iodine, it is also called as grams iodine. It serves as a mordant over here, and it is also put for 1 minute, and then again you wash it. After that, in the next step, what you do, you decolorize the glass slide using alcohol. Okay, acetone or alcohol can be used for decolorization, and decolorization has been done for 10 to 30 seconds, and then you rinse it. And you can see that some of the bacterial cells will lose its crystal violet from there. Okay, and some of the bacterial cells will still remain purple in color. And the next procedure, what you do, onto the same glass slide, you put safranin onto it. Safranin is added and you leave the glass slide for 30 to 60 seconds. Then you water rinse it and you blot dry it. And when you view it, you can see that some cells will appear to be purple in color and some cells will appear to be as pink in color. So all those cells which appear to be purple are being considered to be as gram positive, and all those cells which appear to be as pink in color will appear to be as are considered to be as gram negative in nature. Now, when you saw this picture, you might thought these two as the same. See, they are rods. They are rods. It's a thicker rod, but when the staining procedure was being done, you can see that they are two different microbes because their staining procedures are different. Okay, so let's move on to it. What is the cause of this gram staining reaction like this? In certain case, in certain bacteria, if you go to consider their cell wall structure, okay, see you just look at this. You have a cytoplasm, you have a cell membrane, and outside that you have a, so commonly all the bacteria will have cytoplasm and cell membrane, and outside of that they have a cell wall. And in certain bacteria, in the gram positive cell wall, it is made up of peptidoglycan which is surrounding the cell. Okay, and this peptidoglycan layer has been found to be very thick. Okay, and peptidoglycan, when you stain it with crystal violet, what happened? The primary stain or the crystal violet stain is being taken by the certain bacteria called they are being taken up by some all group of bacteria, but some bacteria do have a higher amount of peptidoglycan, so large amount of crystal violet is taken by it, and even after decolorization by alcohol, they will not lose their crystal violet. But in the case of gram negative bacteria, you just look at that, they have a lesser amount of peptidoglycan, and in addition to the peptidoglycan layer, what happens? It also has an outer membrane. Just remember, it is always I'm just giving you a code. If it is gram negative, though you consider it is gram negative, it has something extra. Just remember that though it is being considered to be as gram negative, it has an additional feature over here. At the cellular structure, the cell wall is having another structure called the outer membrane in addition to the peptidoglycan layer. Now, this green color which is showing over here is the peptidoglycan layer. So, in the gram negative organisms, the peptidoglycan layer has been found to be very thin, and as a result, and it also has an additional outer membrane. So, when you give crystal violet, it takes up crystal violet, but at the same time when you decolorize it, the crystal violet has been lost, and further on it takes up the secondary stain. So that is the reason why all the gram negative organisms will appear to be as pink in nature. It will be more clear to you when you do a practical session in your lab. I guess you got an idea about the gram staining technique right now. So we'll continue with the next differential staining techniques in the coming sessions. Thank you for now.